Good morning. The bibliographic references and the publisher's source article are accessible in the description under the video. If you want to support the dissemination of scientific knowledge, subscribe, and can you give it a thumbs up please? Genetic tracing of market wildlife and viruses at the epicenter of the COVID-19 pandemic. In brief, DNA of mammalian wildlife species susceptible to SARS-CoV-2 was detected along with SARS-CoV-2 and other viruses in environmental samples from animal stalls located at the market epicenter of the emergence of COVID-19. Highlights: common ancestor of SARS-CoV-2 linked to Huanan market matches the global common ancestor. Wildlife mitochondrial DNA identified in samples from stalls positive for SARS-CoV-2. DNA from raccoon dogs, civets, and other wildlife species detected in market samples. Genotypes of potential hosts were reconstructed for retracing animal geographic origins. Summary: zoonotic spillovers of viruses have occurred through the animal trade worldwide. The start of the COVID-19 pandemic was traced epidemiologically to the Huanan seafood wholesale market. Here, we analyze environmental qPCR and sequencing data collected in the Huanan market in early 2020. We demonstrate that market-linked severe acute respiratory syndrome coronavirus 2 SARS-CoV-2 genetic diversity is consistent with market emergence and find increased SARS-CoV-2 positivity near and within a wildlife stall. We identify wildlife DNA in all SARS-CoV-2 positive samples from this stall, including species such as civets, bamboo rats, and raccoon dogs, previously identified as possible intermediate hosts. We also detect animal viruses that infect raccoon dogs, civets, and bamboo rats. Combining metagenomic and phylogenetic approaches, we recover genotypes of market animals and compare them with those from farms and other markets. This analysis provides the genetic basis for a shortlist of potential intermediate hosts of SARS-CoV-2 to prioritize for serological and viral sampling. Introduction: many of the earliest known cases of COVID-19 worked at or visited the Huanan seafood wholesale market Huanan market in the city of Wuhan, a link first made by clinicians at different hospitals throughout the city. One retrospective review of early COVID-19 cases identified 174 patients with onset in December 2019, 32% of whom had an ascertained link to this location within a city of over 12 million. For although some of the early case finding could have preferentially identified market-linked cases, and geospatial analysis of residences of the early cases with no identified link to the Huanan market showed that they lived unexpectedly close to and centered around the market 3-5, even though geographic proximity was not used as a case criterion. 468 additionally, excess pneumonia deaths were first reported in the city districts surrounding the Huanan market 468, and retrospective surveys of Wuhan confirm that a larger proportion of residents contracted COVID-19 in these districts. The genomic epidemiology of severe acute respiratory syndrome coronavirus 2 SARS-CoV-2 shows that there were very few human infections before the earliest ascertained market case with onset on December 10, 2019. 11. The time of the most recent common ancestor TMRCA is estimated to be late November to early December 2019, and the estimated median timing of the primary infection mid to late November. A phylogenetic analysis of the epidemic's size by December 1, 2019 estimated it to be between 183 infections and 02 hospitalizations, 95% highest posterior density intervals. 11. These estimates are consistent with surveillance and retrospective testing, which have found no evidence of substantial community transmission of COVID-19 prior to December 2019. Early SARS-CoV-2 sequences belong to two lineages, denoted A and B, separated by two nucleotide substitutions, C8782T and T28144C. Although the routine of SARS-CoV-2 between these two haplotypes is uncertain. 11. The initial observation that SARS-CoV-2 genomes from cases with direct market contact were lineage B led to the proposal that the market was an amplification event that occurred after lineage or community transmission unrelated to the market. The geographic proximity of two early lineage cases to the market, however, later suggested that this lineage was also present, and this was further confirmed when lineage was identified in an environmental sample from the Huanan market. The linkage of both lineages to the market is consistent with phylogenetic evidence of at least 2 successful zoonotic spillovers of SARS-CoV-2 into humans. 11. The high intensity of contact between humans and animals in markets suggests that once animals infected with a highly transmissible virus arrive in a market, multiple zoonotic events are primed to occur. This was, for example, later observed in a local 2021 COVID-19 outbreak initiated by multiple secondary spillovers from hamsters in a pet store in Hong Kong. In February 2020, China's government enacted a far-reaching ban on the sale of wildlife for human consumption. A similar decision had followed the second emergence of SARS-CoV-1 in winter 2003-2004. Both were intended to limit the further spread of either virus within the animal trade. As with SARS-CoV-2, SARS-CoV-1 was first detected more than 1,000 km from its closest identified bat virus relatives in Yunnan province and was epidemiologically linked to the wildlife trade. Months after declaring the SARS outbreak, closely related viruses were found in musk palm civets, Paguma luvata and common Akun dogs Nycteroutes procyon at still open wet markets, although animals from several markets and farms tested negative for SARS-CoV-1. Farmed civets in Hubei province also tested positive, indicating the spread of SARS-CoV-1 among animals in the province where SARS-CoV-2 later emerged. Zoonotic spillovers in wildlife markets have long been known to present risks for viral emergence. Cross-species transmissions of bat coronaviruses to mammals in the wildlife trade have also been documented among Malayan porcupines Hystrix brachua and hoary bamboo rats Rhizomes pruinosus. Coronaviruses have been reported in musk palm civets, raccoon dogs, and Amma hedgehogs Erinaceus amurensis in wildlife markets, and although the closest relatives of SARS-CoV-2 to date are in bats, other closely related viruses have been found in illegally traded pangolins in Asia. In rural Myanmar, individuals with wildlife exposure had disproportionately high seropositivity to SARS-CoV-2-like viruses, possibly indicating spillover to humans prior to the SARS-CoV-2 pandemic as well. The Huanan market was the location with the most wildlife vendors in Wuhan, a city of over 12 million people with 4 sustained live animal markets. Several vendors were documented to be illegally selling live animals such as raccoon dogs, civets, bamboo rats Rhizomes pruinosus and slash or Rhizomes sinensis, Malayan porcupines, Amma hedgehogs, and Asian badgers Meals leucurus in late fall of 2019. Most wildlife vendors were located in the west wing of the market, which was also where the earliest and the majority of market COVID-19 cases worked. 3. In early 2020, Liu et al. Collected environmental samples from the Huanan market. These samples were analysed by SARS-CoV-2 reverse transcription quantitative PCR, qPCR and metatranscriptomic next-generation sequencing MLNGOs. In addition to environmental sampling, Liu et al. Performed qPCR testing of some mammalian wildlife at the market, but this was limited to live rats, mice, stray weasels, cats and dogs, as well as carcasses of 1 sheep, 2 wild boars, 6 bamboo rats, 6 badgers, 6 muntjacs, 16 hedgehogs and 52 rabbits, most of which are now known as unlikely to be intermediate hosts of SARS-CoV-2. Neither sampling nor qPCR testing of any of the raccoon dogs or civets on sale in the market have been reported, and no serology from any animals or their handlers in the market has been described. However, Liu et al. Reported the genetic detection in environmental samples of several animal genera of potential interest. Here, we analyze the data from the market generated and shared by Liu et al. In the context of all early SARS-CoV-2 sequences using multiple genomic approaches. We first demonstrate that the most recent common ancestor MRCA of SARS-CoV-2 sampled within the Huanan market is genetically identical to the MRCA inferred from the pandemic as a whole. This finding is consistent with the emergence of SARS-CoV-2 within the Huanan market. We next characterize the genetic material from mammals present in market metatranscriptomes at the species level, generating a thorough record of their presence and distribution, including in samples and stalls with SARS-CoV-2. We further identify additional animal viruses, indicating that live mammals sold at the market were indeed shedding viruses there at the time of sampling. Finally, We reconstruct mitochondrial genotypes of putative intermediate hosts in the market for identification of subspecies and their putative geographic origins. Taken together, these analyses provide a precise picture of the genetic signature of wildlife mammals, their viruses, and SARS-CoV-2 present at the Huanan market as the COVID-19 pandemic began. Results: SARS-CoV-2 genetic diversity linked to the Huanan market is consistent with market emergence. If the Huanan market was the site of origin of the transmission chains that led to the COVID-19 pandemic, then the common ancestor of market-associated viral genotypes should be equivalent to the common ancestor of the pandemic, given appropriate sampling. To test this hypothesis, we assessed intrasample variation of the SARS-CoV-2 environmental genomes from the Huanan market, assigned market sequences to virus lineages, and performed phylogenetic inference to compare the genetic diversity of SARS-CoV-2 within the market to its genetic diversity globally. As detailed below, we found that the MRCA of market-associated genomes was equivalent to the MRCA of the larger pandemic. 4 near-complete SARS-CoV-2 genome sequences were recovered from environmental samples collected on January 1, 2020. The samples, labeled A20, B5, F13, and F54, were all collected in the west wing of the Huanan market, where most wildlife vendors were located. For we first confirmed that the sequence from sample A20 was indeed lineage A, whereas samples F54, F13, and B5 were lineage B, as previously reported. The sampled reads strongly support the OB consensus genotype at the lineage-defining sites, indicating that none of these sequences are a mixture of lineages, and B, the A20 consensus genome, has an additional 2 substitutions, G26N262T and C6N145T, separating it from the lineage root, but with the 6N145C reference allele present at 23% frequency, supporting the presence of a haplotype that was in private substitution diverged from the MRCA of lineage A, although the exact timing of the shedding of virus present in this lineage sample remains uncertain. A map shared during the World Health Organization who convened global study of origins of SARS-CoV-2 China part hereof who mission noted a suspected case in the stall where it originated, with onset on or before December 15th. This case was not included on the final map provided in the WHO mission report, but the report recommended follow-up mapping and review of potential early clinical cases. 4 low-coverage sample F46 has 2 duplicate reads, covering the 8.782 position and matching lineage as C8.782T, but this sample has no coverage at position 28.44, the other lineage-defining position, so this sample could not be fully determined to be a second lineage sequence sampled at the Huanan market. To compare the genetic diversity of SARS-CoV-2 sampled within the market to SARS-CoV-2 genetic diversity globally at the onset of the pandemic, we performed phylogenetic inference using the Bayesian evolutionary analysis sampling trees program, BEAST, C-star methods, using the sample collection date of January 1, 2020, for market environmental genomes, and by incorporating recently published genomes from COVID-19 cases from early 2020, we inferred the ancestral haplotype and TMRCA of the SARS-CoV-2 pandemic N equals 863 genomes collected by February 14, 2020, we considered both unconstrained routing and routing constrained by the inferred genome of a recombinant common ancestor RECCA. 11 in both cases. These analyses strongly favoured MRCA of SARS-CoV-2 only as either the LB haplotypes with no additional substitutions or an intermediate between the two, as did previously published results. The alternatively proposed a plus C18060T and a plus C29095T 23 haplotypes were strongly rejected as being ancestral, including, when routing with the inferred RECCA, the MRCA of market-linked genomes on all 4 plausible tree topologies was therefore concordant with the common ancestor of the entire pandemic. We inferred a median TMRCA of December 09, 1995%, highest posterity density HPD at November 11 December 20, for the SARS-CoV-2 pandemic with unconstrained routing, and of December 0395% HPD November 04 December 16 with RECCA constrained routing, consistent with prior analyses, that the MRCA of SARS-CoV-2 linked to the Huanan market is equivalent to the MRCA of the pandemic establishes that the timing of the origin of the market outbreak is genetically indistinguishable from the timing of the origin of the pandemic as a whole. The presence of both lineages A and B at the market and the spatial association of early lineage cases with the market 2-3 were results directly predicted under the hypothesis that SARS-CoV-2 first emerged in the human population at the Huanan market. Increased SARS-CoV-2 positivity rate in and near a wildlife store at Huanan market. The Huanan market was sampled on multiple dates at the start of 2020, with different sampling trips having different purposes. On January 1st, a market was sampled widely, with an emphasis on stalls associated with human cases. 515 samples were tested, with SARS-CoV-2 qPCR positive, and mNGS sequencing results for 25 of these positive samples were shared. On January 12th, 10 samples per stall were taken from 7 wildlife stalls. 3 were positive by qPCR for SARS-CoV-2, and this time all 70 samples including negatives were sequenced. Additional samples were collected from drains, sewage, stalls and warehouses after these 2 first dates until March 2020 to determine whether SARS-CoV-2 positivity was associated with specific stalls in the Huanan market. We conducted a spatial relative risk analysis of SARS-CoV-2 qPCR positive samples collected on January 1st and 12th, comparing the distribution of the qPCR positive to qPCR negative samples. The rate of qPCR positivity was unevenly distributed within the Huanan market, with increased positivity in the southwest section. Several clustered stalls in this section had a higher positivity rate than the average stall sampled in the market. One stall, wildlife stall A, stood out, with a 30% qPCR positive rate. 3 of its 10 samples collected on January 12th, a cart, a hair/feather removal machine, and a sample collected from the ground were qPCR positive for SARS-CoV-2. 6 of the 70 January 12th sequenced samples contained SARS-CoV-2 mNGS sequence reads, which included the 3 qPCR positive samples from wildlife stall A, 2 qPCR negative samples also from wildlife stall A, and 1 sample from the interior of a freezer in nearby wildlife stall B. Therefore, both qPCR testing and mNGS identify SARS-CoV-2 RNA in and around wildlife stall A. Although SARS-CoV-2 read counts are low, this is consistent with the precedent for untargeted environmental sequencing of SARS-CoV-2 in which viral RNA can be nearly undetectable even in PCR-positive samples where the overwhelming majority of sequences are microbial. Later sampling provided further support for wildlife stall as a SARS-CoV-2 hotspot within the Huanan market. Of 60 samples from the underfloor drainage system of the Huanan market that were collected on January 27th and 29th, only 4 were SARS-CoV-2 qPCR positive, including the drain directly in front of wildlife stall A. 17 more drain samples were collected on February 9th and 15th, with 3 testing qPCR positive. 1 of these was again in front of wildlife stall, and the other 2 are from downstream locations that may have received runoff from this same stall. Taken together, there are 3 independent spatial signals that identify SARS-CoV-2 positivity associated with a specific wildlife stall or in a section of the market with markedly higher environmental SARS-CoV-2 positivity. The nearby wildlife stall B was also repeatedly resampled after January 12th and tested qPCR positive multiple times. In February, the offsite warehouse associated with this stall was sampled, and 5 of 12 samples tested positive for SARS-CoV-2 by qPCR. Of the 16 samples collected from wildlife stall B on January 1st and 12th, SARS-CoV-2 was detected only by mNGS in the January 12th freezer sample, indicating lower positivity than wildlife stall A. Outside of these wildlife stalls, several of the other SARS-CoV-2 positive stalls sampled on January 1st were associated with the known human cases in the market. These samples most likely reflect human shedding of SARS-CoV-2 in these other locations throughout the market, which was a site of ample human-to-human transmission. 3 As time of sampling progressed after the market's closure, there was a noticeable decrease in SARS-CoV-2 viral abundance, indicating environmental viral RNA decay throughout the market over several weeks. As most wildlife stall samples were collected 11 days after the first sampling, a reduced capacity to detect SARS-CoV-2 in wildlife stall samples on January 12th would then be expected due to ongoing decay of viral RNA in the environment. Mammalian wildlife species detected in 5 SARS-CoV-2 positive samples from a wildlife stall. Environmental samples with viral RNA can also contain genetic evidence of the mammalian hosts that shed that virus. We developed and benchmarked a highly specific metagenomic pipeline for quantifying the abundance of animal mitochondrial DNA, mtDNA, in environmental samples, by stringently mapping reads to a dereplicated database of eukaryotic mitochondrial genomes and filtering hits by their breadth of coverage. The 5 SARS-CoV-2 positive samples from wildlife stool that contained mtDNA from raccoon dogs, oribamboo rats, dogs Canis lupus familiaris, and European rabbits Oryctolagus cuniculus, and my hedgehog and Malayan porcupine mtDNA was present in 4 samples. Rhesus Mungic Muntiacus revisi and Himalayan marmot Marmota himalayana mtDNA was found in 3, and 1 sample contained masked palm civet mtDNA. Of these species, raccoon dogs, rabbits, and dogs are documented as susceptible to SARS-CoV-2, with raccoon dogs experimentally confirmed as capable of transmission and in vitro evidence of civet susceptibility. Nearby to Storway, other SARS-CoV-2 positive samples also contained wildlife mtDNA, including a garbage cart where raccoon dog mtDNA was detected and a stall with bamboo mtDNA. Although all 5 positive samples from stall contained human mtDNA, humans were not most abundant mammalian species present in any of these samples, excluding the mitochondrial 16s and 12s rRNA regions, which could be differentially impacted by potential rRNA depletion performed on these samples, not change these results. Further, our results show that wildlife mtDNA detection was co-localized with the reported locations of wildlife stalls. In contrast, human mtDNA was distributed throughout the market, consistent with the market being a general place of human activity. Genetic material from raccoon dogs and hoary bamboo rats was frequently found across the wildlife stalls, constituting 2 most commonly detected mammalian wildlife species. Genetic material from masked palm civets was more rarely detected, being present in 5 samples from 4 stalls. Some wildlife species, such as Nutria mycostocoipus, red foxes vulpes, and Arctic foxes vulpes, lagopus, were detected only in samples and stalls that tested negative for SARS-CoV-2. To confirm species identification, we generated de novo contig assemblies and performed blast against a custom whole genome sequencing WGS database made from available genome assemblies of species known to be at Huanan market. The blast results identified human and wildlife species consistent with those described above, of the 18 species reported by Zhao et al. To be present in the 4 Wuhan city markets they surveyed. We confirmed genetic signatures 11 at the species level at the Huanan market and an additional 2 at the genus level. Siberian weasel Mustela sibirica was rare in the market and was absent from any of the January 12 wildlife stall samples. We did not detect mtDNA sequence reads from American mink Neogale vison, red squirrel Cirrus vulgaris, Pallas squirrel Callicirrus erythraeus, complex-toothed flying squirrel Trogopterus xanthipes, nor Asian badger in any of the environmental samples. Notably, none of these, except Asian badgers, were observed at the Huanan market specifically in November 2019. Additionally, while Zhao et al. Reported sales of Chinese bamboo rat R. sinensis, we identified abundant mtDNA from the hoary bamboo rat R. pruinosus, with only 1 sample containing trace amounts of R. sinensis mtDNA. This discrepancy is likely due to the species having been identified by Zhao et al. Visually based on physical morphology, without genetic confirmation. We found no evidence for the presence of any bat or pangolin genetic material, the 2 known hosts of Sarbecovirus relatives of SARS-CoV-2, in the Huanan market. In contrast, the presence of mtDNA of Myotis bats was previously reported. To check this, we replicated the methodology of Liu et al. By mapping reads to the barcode of Life Data System COX1 gene database, which identified 8 reads that mapped to any Myotis sequence with no mismatches. A blast analysis confirmed that all were non-specific matches and therefore uninformative. This indicates that neither live bats nor pangolins are likely to have been present in the sample stalls of the Huanan market during the time period relevant to the emergence of SARS-CoV-2. Prior studies calculated correlations of SARS-CoV-2 and animal sequence redundancies in market samples, concluding that SARS-CoV-2 was negatively correlated with mammalian wildlife species. Such approaches are limited because metagenomic sequencing counts are relative compositional and further subject to temporal trends, spatial trends, and extraction biases that can all confound correlational interpretations in environmental samples. These approaches are also challenged by the consideration that animal viral shedding would precede human viral shedding in a zoonotic scenario, and that most wildlife stools were sampled 11 days after stools with suspected human COVID-19 cases. As a result, environmental SARS-CoV-2 RNA from non-human hosts could have had more time to decay than SARS-CoV-2 RNA from human infections. In addition, SARS-CoV-2 detected in non-wildlife stools was very likely shed by humans, rendering univariate correlations, including these samples, inappropriate. In terms of experimental design, the overall sampling scheme of the market sampling was also imbalanced. All wildlife stool samples from January 12 were sequenced regardless of their qPCR positivity, while other sequenced samples were predominantly qPCR positive and from elsewhere in the market. Therefore, species present in wildlife stools are disproportionately overrepresented in the sequence negative set, and this sampling design will cause wildlife species to artifactually appear negatively correlated with SARS-CoV-2. As all wildlife stool samples collected on January 12 had been sequenced regardless of their SARS-CoV-2 positivity, we conducted a correlational analysis of relative species abundances in these samples n equals 70, as this could represent a balanced dataset for informing which host had shed the virus detected therein. Across these wildlife stool samples, there was no significant correlation between human mtDNA and SARS-CoV-2 RNA R equals 0. 13. 95% confidence interval CA0. 090. 34. Similar to the average mammal R equals 0. 08. 95% psi0. 12 29. Sequence rare abundances of Malayan porcupine R equals 0. 45. P0. 001. False discovery rate FDR equals 5%, and Himalayan marmot were significantly correlated with SARS-CoV-2 after multiple hypothesis correction R equals 0. 34. P0. 033. FDR equals 5%, reflecting their increased detection in wildlife stool A. However, temporal trends and compositional effects in metagenomic sequencing data also influence correlations, further challenging their interpretation. As previously described, a correlational analysis would be unlikely to provide reliable insights into whether any particular species was or was not infected by SARS-CoV-2 within the market. Wildlife stools and SARS-CoV-2 positive samples contain other mammalian viruses associated with the animal trade. The presence of animal viruses with predictable host ranges can provide evidence of animals productively infected with viruses in the Huanan market at the end of 2019. By mapping sequencing reads to a custom database of human and animal viruses with stringent filtering, we identify several mammalian viruses present in the market. Human-specific viruses were rare, even at a threshold of 1 rid per sample. We found human coronavirus 229E in one stool and human respiratory syncytial virus subgroup B in another. Other detectable human viruses were double-stranded dsDNA viruses, such as human polyomavirus, human papillomaviruses, and human herpesviruses. We also detected several other mammalian viruses within the market. In SARS-CoV-2 positive wildlife stools, we identified close relatives of viruses reported to infect the wildlife species also detected in these samples. Of these viruses, close relatives of a raccoon dog amniparvovirus, a bamboo rat betacoronavirus, and a civet copuvirus were sufficiently abundant to reconstruct mostly complete genome sequences from samples across the market via a mapping consensus approach. All 3 viruses were predominantly found in wildlife stools with MNGS evidence of their putative hosts, and in some cases they were identified in samples from nearby locations as well. We inferred maximum likelihood whole genome phylogenetic trees incorporating these viruses and their known relatives, with IQ tree 264 under a GTR plus F plus G4 substitution model. In each case, the virus present in the market was most closely related to reported viruses shed by a singular host species, indicating a predictable host specificity. The two closest relatives of the civet cobbyvirus we detected were from sequences identified in samples from market animals from Sichuan and Guangxi provinces, and the bamboo rat beta coronavirus was a close and recent relative of a virus identified in bamboo rats on the Guangxi farm in 2019. These findings suggest some movement of infected animals from southern China to Wuhan, a trade conduit that could have also led to the emergence of SARS-CoV-2. This result is also consistent with reports that Huanan market vendors sourced bamboo rats from Guangxi and Yunnan provinces. For movement of animal viruses such as these via the wildlife trade recapitulates the likely dispersal of SARS-CoV-1 from Yunnan to Guangdong and Hubei provinces. Additional viruses found at the market included polyomaviruses, hedgehog coronavirus HKU31, and skunk adenovirus PB1, which has a broad host range. Further, we identified canine coronaviruses closely related to viruses sampled from dogs, farmed and captured raccoon dogs, and farmed red foxes. These viruses may be expected to infect multiple canine species present in the market. All 8 genome segments of influenza virus subtype H9N2 were detected together in a sample from one SARS-CoV-2 negative wildlife stall, and with sparse detection 10 reads in 2 other samples from the same stall and 2 samples from adjacent wildlife stalls. Manual inspection of the reads identified the presence of the G57-like internal gene cassette, the predominant H9N2 genotype in China, also shared with zoonotic H7N9 viruses from China. Read mapping to HA genes identified 2 different HA lineages, G1 eastern sublineage and Y280/BJ94, mixed in the same sample, as well as the presence of an N2NA gene, indicating the presence of 2 H9N2 lineages colocalized in the market. Phylogenetic analysis of the HA gene indicated that the G1 virus was most closely related to sequences collected in Guangdong in 2018. Spillover of avian H9N2 had been reported in civets. In another recent survey of market animals in China, human zoonotic cases of H9N2 have occurred. Hence, SARS-CoV-2 was not the only virus with zoonotic potential present in the Huanan market at the end of 2019. These results further add to the evidence for the presence of live animals at the market and establish it as a place where potential wildlife hosts of SARS-CoV-2 were actively shedding other viruses. The construction of mitochondrial genotypes of potential intermediate host species of SARS-CoV-2 within the Huanan market. Genotypic differences within species can be valuable for identification of the subspecies and the geographic origin of individual animals present at the Huanan market. To facilitate subspecies identification and ascertainment of the likely geographic origin of animals in the Huanan market, we reconstructed mtDNA consensus genomes from wildlife stall samples. We used a reference-guided mapping approach to obtain partial to near-complete mitochondrial consensus genomes of several mammalian wildlife species. We obtained 33 consensus mitochondrial haplotypes from separate swabs that were 50% complete for 7 abundant wildlife species: raccoon dog, masked palm civet, hoary bamboo rat, amma hedgehog, Malayan porcupine, greater hog badger Arctonix calaris, and the Himalayan marmot. 10 of these mitochondrial genomes were 90% complete. We further identified the consensus single nucleotide variants isenthius for these genomes and found that, on average, they diverged 0.57% from the reference genome for each species, minimum of 0.16%, maximum of 2.1%. To investigate the geographic origins of the raccoon dogs in the Huanan market within the context of the animal trade, we analyzed their mtDNA genotypes. In Asia, the raccoon dog's current range extends from Vietnam to Russia, and 4 subspecies are recognized: 1N. P. Procyonides found throughout China, 2N. P. Corensis in the Korean Peninsula, 3N. P. Orsts found in southern China and Vietnam, and 4N. P. Usheriensis, a subspecies found in northeastern China and Russia and farmed in large numbers in this region for its thick fur. Given the natural range of the closest known bat sarbecovirus relatives of SARS-CoV-2 in southern China, northern Laos, and Vietnam, raccoon dogs from northern China would be an unlikely conduit for SARS-CoV-2 into Wuhan. We performed phylogenetic inference on the mitochondrial CYTB gene, as reference genes have been reported for wild raccoon dogs Hubei Province 2020 and Vietnam 2013, and multiple raccoon dog subpopulations can be distinguished using the SITB gene, because market environmental samples can contain DNA shed by unrelated but co-housed animals of the same species. We used a metagenomic SNV-based approach instead of relying solely on the consensus genomes. To compare animals in the Huanan market to those from other markets, we identified SNVs for mitochondrial genomes from an independent set of samples of pooled raccoon dog samples collected from markets in 5 cities to the north of Hubei Province during 2020. Notably, raccoon dog samples from the other markets to the north of Hubei Province were characterized by the presence of SNVs on 2 branches to N.P. Usheriensis and N.P. Procyon sampled on fur farms and in the wild in northern China and Russia. These SNVs were absent from the Huanan market. Instead, SNVs from 2 genetically distinct raccoon dog populations sampled in 2020 and collected in the wild in Hubei in 2020 were present. SNVs associated with 2 raccoon dog haplotypes collected in Vietnam in 2013 were absent from all samples. This suggests that the detected raccoon dogs in the Huanan market in late 2019 were not N.P. Usheriensis and were from at least 2 populations, both genetically distinct from those raised on fur farms or sold in markets in cities in northern China. It is unknown how far south the wild and farmed range of the subpopulations detected here extends due to a lack of genetic data for N. Procyonids on the farms found in central or southern China. These data are consistent with the geographic origin of the raccoon dogs in the Huanan market in central or southern China, from which a viral transmission chain within the animal trade could have arisen after a spillover from a bat reservoir south of Wuhan. Discussion. Extensive epidemiological evidence supports wildlife trade at the Huanan market as the most likely conduit for the COVID-19 pandemic's origin. Herein, we both describe metagenomic evidence for identification of the plausible intermediate host species at the market and analyze metagenomic and phylogenetic evidence that further supports the hypothesis of zoonotic emergence at this location. Although the species identity of an intermediate host between the likely Rhinolophus spp. Horseshoe bat reservoir of SARS-CoV-2-like coronaviruses and humans remains unknown. Our analysis informs this open question by determining the mammalian species present in the market with species and subspecies resolution. These results show that multiple plausible intermediate hosts of SARS-CoV-2 were present at the exact site within Wuhan to which COVID-19 was first epidemiologically linked. It is not possible to conclude which of these species may have been infected and or introduced the virus to the market from these data alone. Nonetheless, our analysis provides a short and actionable list of species with genotypic details. Of the wildlife species detected in SARS-CoV-2 positive environmental samples 4 have previously been implicated in bat coronavirus cross-species transmission through the animal trade of raccoon dogs, masked palm civets, Kori bamboo rats, and Baleine porcupines. Among the potential intermediate hosts present in the Huanan market, raccoon dogs are known to be susceptible to SARS-CoV-2, to shed high titers of virus, and to be able to transmit. The common raccoon dog was the most abundantly detected animal species in market wildlife stalls sampled on January 12th and in the wildlife stall with the most SARS-CoV-2 positive samples. The majority of civet mtDNA detectable in the market was from wildlife stall A, and while the susceptibility of civets is unknown, Paguma larvata cell lines are susceptible in vitro. The susceptibility of bamboo rats, Malayan porcupines, and amma hedgehogs remains unknown, and these species should be prioritized for susceptibility testing. Of the other species in the market, marmots may be an unlikely conduit for SARS-CoV-2 considering that their usual range is at very high elevations, and although muntjac deer are closely related to cervids susceptible to SARS-CoV-2, the species has a truncated ACE2 gene without a signal peptide sequence, which may suggest a lack of susceptibility. The Siberian weasels, foxes, and greater hog badgers detected in the market are closely related to species known to be susceptible to SARS-CoV-2, but these species were very rare or absent in SARS-CoV-2 positive samples and stalls. Other market mammals are known to be susceptible to SARS-CoV-2 but do not represent a significant transmission risk, including dogs, rabbits, and boar, as infected animals do not produce or shed virus at high titers. Multiple lines of evidence are consistent with the infection of wildlife animals with SARS-CoV-2 in the Huanan market. Animal carts, a cage, a garbage cart, and a hair/feather removal machine from a wildlife stall tested positive for SARS-CoV-2, and there was more DNA from mammalian wildlife species in these samples than human DNA. The surrounding stalls also had relatively higher rates of SARS-CoV-2 positivity, and drains adjacent to and downstream of this wildlife stall tested positive for SARS-CoV-2. Finally, there were several other viruses known to infect wildlife in these samples. These data indicate either that the animals present at this stall shared the SARS-CoV-2 detected on the animal equipment, or that early unreported human cases of COVID-19 shared virus in the exact same location as the detected animals. Under a zoonotic scenario, viral shedding by infected animal handlers is expected to occur after shedding by the animals they were infected by. Therefore, human viral shedding post-zoonosis could plausibly also explain these data. In such a case, the animals brought into the stall are therefore still the most likely candidates for the zoonotic source. Although either animal or human viral shedding is consistent with these data, only a zoonotic origin of SARS-CoV-2 directly predicts co-detection of SARS-CoV-2 and wildlife genetic material. It has been proposed that humans could have introduced the virus into the Huanan market. It is most likely that there were human infections of SARS-CoV-2 earlier than the first documented and hospitalized market cases, including unascertained market cases or contacts thereof. However, the detection of lineage B and lineage are both within and indirectly geographically linked to the Huanan market implies that SARS-CoV-2 most likely emerged there or its supply chain before the TMRCA, by which time there would have been an estimated median of just 3 people infected 95%, HBD, 118. Any hypothesis of COVID-19 emergence as to explain how the virus arrived at one of only 4 documented live wildlife markets in a city of Wuhan's size at a time when so few humans were infected. 3 human introductions linked to the animal trade offer one explanation for this, and the introduction of the virus by an animal trader or farmer cannot be excluded, but these hypotheses are challenged by phylogenetic evidence for multiple spillovers. The introduction by an animal trader infected by animals upstream of the market is further challenged by the probability that transmission chains dependent upon a single human would likely go extinct, while sustained interface between infected animals and humans in a market is more likely to result in the establishment of an epidemic. The detection of SARS-CoV-2 RNA in the Huanan market in January 2020 could plausibly reflect deposition several weeks before sampling, compatible with estimated dates of the first human infections. SARS-CoV-2 RNA has been detected on indoor surfaces for prolonged periods of up to several weeks, and the temporal signal of viral rRNA and animal mtDNA decay in the Huanan market samples themselves offers further support for these timescales. Although the earliest zoonotic events of SARS-CoV-2 most likely occurred in late November 2019, infected cohouse animals could be expected to shed virus for weeks longer. SARS-CoV-2 genetic material detected in the Huanan market may be a remnant from either animal or human sources during any time in that period, and this could vary both within and among samples because of differing environmental conditions and or decay rates of various nucleic acids. Focused genetic and serological sampling of raccoon dogs and the other mammalian species reported here throughout Southeast Asia and southern China can shed light on the animal trade networks that may have led to the emergence of SARS-CoV-2, as previously recommended. For serological testing of the oldest animals, for instance, the breeding stock in source farms might provide additional information of transient circulation, as has been observed in mink farms infected with SARS-CoV-2. Future studies to clarify the susceptibility status of all of these species using in vitro approaches and live animal infection experiments should also be prioritized. The limited viral and serological sampling of these species in Southeast Asia and southern China indicates that the wildlife trade directly before the COVID-19 pandemic is highly undersampled or underreported. Retrospective studies should be performed, where possible, testing the species described here throughout the animal supply chains of Southeast Asia and southern China, through which in all scientific likelihood the COVID-19 pandemic emerged. Limitations of the study. Because the environmental metagenomic data used in this work cannot directly link viruses to their hosts in samples that contain DNA or RNA from multiple plausible host species including humans, our analysis cannot conclusively identify which species may have shed SARS-CoV-2 in different samples from the Huanan market. Similarly, the exact timing of when viral or host genetic material was shed in the market environment cannot be directly estimated. The samples sequenced from wildlife stalls analyzed here were sampled 11 days after several other stalls in the market, and SARS-CoV-2 sequencing read counts were low in both qPCR positive and qPCR negative samples from these stalls. The amount of degradation that occurred between deposition and sampling affects the relative abundances of genetic material from different species and cannot be quantified. In addition, metagenomic sequence abundances are influenced by extraction biases specific to the species, virus, and type of genetic material, and by the technical specificity and sensitivity of different computational approaches. It is also challenging to distinguish very closely related animal species or subspecies without reference sequences, particularly in samples with very low coverage of the target. Finally, the publicly available genomic and epidemiological data from the start of the COVID-19 pandemic remain incomplete, and future data from this time could shed further light on hypotheses regarding its emergence,